This recorded session is to cover our remaining topics on primary metabolism in microbes. So we'll tackle three more um, major pathways or um, biosynthetic um, pathways for the production of primary metabolites. So, we ended last time with nucleotide biosynthesis. So, for bacteria, purine and pyrimidine biosynthesis are processes that are not salvageable, meaning they are produced from the ground or from the raw materials to the final molecules. So bacteria synthesize nucleotides de novo. So there are the pathway for producing purines and pyrimidines in bacteria have been well studied, and what has been found out is that um, primary processes happen at the level of the cell. So the bacterial species do not really get or salvage their nucleotides from other species or from the environment. Okay, so we're reminded about the importance of nucleotides. Nucleotides are involved in multiple functions. Some are energy currency, some are energy molecules, particularly ATP and GTP. Of course, very important, or the most important function is the fact that nucleotides are building blocks of nucleic acids, both DNA and RNA. And some nucleotides are also important for cell signaling, such such as those that are involved during processes that require signal transfer. Um, to begin looking at the process of nucleotide synthesis, for the case of purines, adenine and guanine, there is a base purine molecule. No? So the base purine molecule, as shown here, is inosine 5 monophosphate. So this base purine molecule serves as a branch point for the production of ATP and GTP. No? Then for pyrimidine biosynthesis, the case is quite different, but what marries the two pathways is a very important molecule, um, PRPP, so 5-phosphoribosyl alpha 1 pyrophosphate. So this is the base molecule of purine and pyrimidine biosynthesis. Not really base, but more of a branch point where the both pathways converge into a single network. No? So before we discuss this, I'd like to go back to this slide. No? So it has been for the case of microbes, it has been well theorized that nucleotide synthesis can be an important element in the survival and the pathogenicity of bacteria. That's why we've been looking into targeting the DNA or RNA molecules of pathogenic bacterial species, as these are the blueprint of processes or expression of virulence factors. Um, Now, um, at the core of the pathways, so here are the processes that are involved. No, so for purine biosynthesis, a key molecule is PRPP. So let's remember this. 5-phosphoribosyl alpha 1 pyrophosphate. It's a multiple phosphorylated compound. It's a common precursor to purine and pyrimidine biosynthesis. No? There are several enzymes that convert PRPP into multiple intermediates. So here, this is a graph, or this is a, do we have it here? No, I didn't place it. But anyway, so there are several enzymes that convert PRPP to intermediates before IMP is actually produced. No? So I'd like you to be familiar on these, but don't worry about memorizing them, okay? Or memorizing the name exactly. So some important things that I just want to emphasize include, so from steps 2 to 7 there are multiple conversions that take place. Take note of the enzymes that are involved, the one in parenthesis. So this is in order of the intermediate conversions. So we have upper F, D, N, T, L, S, Q, M, K, and E. So from steps 1 to 7, these are just conversions from the from PRPP, the starter molecule, down to 5-phosphoribosyl 5-amino-4 imidazole carboxylate. Okay. Now step number 8 is what I want us to focus, no? Because in this step, the catalysis of the base molecule for ATP and GTP synthesis is now going to finally happen. Okay? So three enzymes that are universally conserved among bacteria per C, B, and H, they all work together for the transition of our 5-phosphoribosyl 5-amino-4 imidazole carboxylate to three more intermediates respectively. And in the last step, so this one, no, I don't have it here. But in the last step, so the final enzyme that will carry out the process of producing IMP, inosine 5 monophosphate, is per H. Okay, so per H is catalyzed from um, the final intermediate processed by oops, the final intermediate, so FAICAR, to inosine 5 monophosphate. So per H catalyzes that reaction. No? Then later on, IMP can be fed into different or to separate branches that can yield or that yield the synthesis of GTP or ATP. Okay? So let's remember. And okay, so I think that's all for purine biosynthesis. Now, for pyrimidine biosynthesis, it's important. Unlike as I mentioned a while ago, do I have it? As I mentioned a while ago, a common a node shared by purine and pyrimidine biosynthesis is PRPP. No? Okay, so let's look into the steps. So the process begins for pyrimidine biosynthesis. The process begins with the formation of carbamoyl phosphate, which is also both for ano no for pyrimidine and amino acid synthesis, amino acid production, particularly arginine. No? The enzyme involved is enclosed in parenthesis. Then carbamoyl phosphate is converted to carbamoyl aspartate. Okay. Carbamoyl aspartate can then be modified to dihydroorotate. That's via the enzyme PYR-C, which is the first point where the ring structure is formed. No? The next reaction after conversion of carbamoyl aspartate to orotate um, uh, is the production of double bond. So look into the close ring, essentially forming the first pyrimidine compound that's carried out by the enzymes PYR-K and PYR-D. Okay, and then orotate is converted to orotidine 5 phosphate by the action of PYR-E in the presence of PRPP. So step number 5 is something that I want you to focus on. Okay, so PRPP, in the presence of PRPP, orotate will now be converted to orotidine 5-phosphate. So here, OMP. So PRPP alongside with orotate in the presence of enzyme PYR-E will result in the production of UMP, uridine 5 monophosphate. So UMP is converted or produced by the action of PYR-F, no? And then later on, uh, um, the last step, which is involves the irreversible decarboxylation of orotidine 5-phosphate to UMP. Actually, the process is entirely I wouldn't say yes. It's irreversible in the sense that these are phosphorylated intermediates already that cannot go back to the orotate conversion or orotate conformation anymore. So UMP now can feed towards the process or the branch point for the synthesis of your pyrimidines, of your cytosine, uracil, no? Okay, and thymine. No? Okay, right. So that's everything for nucleotide biosynthesis. Now let's cover vitamin biosynthesis. So vitamins have, in the case of microbes, are also categorized into two. So there are fat-soluble ones and water-soluble ones. Fat-soluble vitamins and factors are usually components of the cell membrane, while water-soluble ones act as coenzymes important for chemical transport. No? Okay. As an example, I want us to look into the synthesis of B12, cyanocobalamin. So it's a vitamin synthesized by bacteria, and for the case of vertebrates, it's important so that we avoid, or for the case of animals, it's important to be consumed or to be part of our diet so that we don't get anemia. No? Now, at the microbial level, at the microbes producing them, so there are several pathways for the production of B12. No? So there are de novo pathways that synthesize cobalamin from simple precursors, so from the raw materials up to the very intricate B12 molecule that you can see here. And there are also salvage pathways. So salvage pathways that recycle or modify the corinoid precursor from the environment. So here you can see the central corinoid molecule, the one associated with cobalt. So this is a very complicated molecule. So it's difficult to synthesize. Therefore, uh, microbes producing B12 also have the tendency to just salvage corinoid rings from the environment because it's a big molecule. It's very complicated in structure, so it's difficult to synthesize it de novo. Okay. Right. Now, a lot of microbes have been characterized in terms of their B12 production. So some of them are able to carry out both de novo and salvage pathways. Others are only confined to one. No? So here, this table shows you or shows to us some examples, no, of microbial species able to carry out cobalamin synthesis and whether they use de novo or salvage or both pathways. Okay. Now, more about the process of B12 synthesis. In terms of regulation, okay, so there are two possible routes for cobalamin synthesis. There is an oxygen-dependent one and another that does not depend on oxygen. But primarily, these two routes can be taken reliant to when the cobalt atom is inserted in the corinoid molecule. No? So first, there is cocylation via hydrogenobirenic alpha C diamide with the complex. No? Oxygen is required during the contraction of the coronoid ring for this step. So in this example, this has been observed in a particular species of Pseudomonas denitrificans. Then another where oxygen is not required in the case of, or as seen in Salmonella type murium, cocylation happens via a different element or via a different factor, precorin 2 with CBIK. So oxygen is not required for this particular species. Okay, again, the regulation of the ring formation or the ring contraction for case of B12 can be oxygen dependent or not. No. Now, I've already shared a bit ago about how salvage pathways are also utilized by bacteria in the synthesis of B12. No, it's a cost-effective pathway. So for both bacteria and archaea, it's very time and energy saving. No, so in gram-negative bacteria, there are the corinoids from the environment. They can be translocated to the cell via ATP binding cassette systems, the ABC transport system. So the you might probably remember from your medical microbiology for key proteins comprising the ABC transport system, BTUC, D, and F. All of they are permeases, ATPases, and periplasmic binding proteins. No, so these ABC transport system components allow the intake or process the intake of corinoids from the environment inside so that they can be used for cobalamin synthesis. Okay, then BTUB is a TANB dependent transporter rather, that is on the outer membrane of gram-negative bacteria. They deliver the corinoid first to the periplasmic space received by the protein BTUF. Then the latter will deliver it to the CND complex located in the inner membrane before it can finally go inside the cytoplasm of the gram-negative bacteria. Okay? So in this example, or in this slide, we're showing you how pathways can be optimized. No, this is in no, in light of B12 biosynthesis. So I ask you to do a further reading on this article. So since B12 is a commercially marketable or significant product, no, it the bioproduction of B12 by microbes has been optimized or has been exploited by biotechnologists. No, so the cycle shows us the process of designing, building, testing, and learning how an industrially significant microbe can be further optimized and explored for the production of an important bioproduct. No? I've been discussing this already since over module number 1. You can just go back to the concepts that we have discussed so far. But you can take a quick read now or skim on this article. This is applicable to this one, vitamin B12 production. No? 0631, yes. Yeah, okay. All right. Now let's talk about the last topic on primary metabolism, polysaccharide synthesis. Okay. So for the most part, polysaccharides exist as conjugates. They are conjugated to a lipid molecule. No? So in bacteria, the polysaccharides are not existing as simple molecules. They are complex ones conjugated with lipids, usually in gram negatives. So the everything outside the cell, at the level of the membrane. So we have The LPS, the capsule, exopolysaccharides, lipid A. So all those things that are polysaccharide rich and are at the same time conjugated to lipid molecules. At the functional aspect, so most of these complex polysaccharides are acting as signaling molecules, important for molecular recognition, important for cell protection. They may also modulate response of the host in the context of a pathogenic bacteria trying to invade or to invade the host and evade the immune system. So example is lipid A, no? Now, for polysaccharide synthesis, a good example I want us to focus on is the O antigen, no? LPS O antigen of gram negatives. Now, the initiating event is the synthesis of a sugar lipid conjugate. So here, figure A. So a sugar lipid conjugate. So you can see here a sugar tail that's being conjugated to a lipid. No, so the process can be mediated by different enzymes. There are two examples, no, of enzymes that carry out that conjugation of sugar lipid of the sugar lipid components. So what happens is there is a transfer of sugar 1 phosphate to a carrier to a base to not a base but a root molecule and decaprenyl phosphate. Okay, and the enzymes that carry out this initiating process are known as follows: PHPT and PNPT. So PHPT, polyisoprenyl phosphate hexose 1 phosphate transferase, PHPT, while PNPT is polyisoprenyl phosphate and acetylamino sugar 1 phosphate transferase. So both are transferases. No? Okay, so as an example, let's have here the O antigen. Okay, so at the outer membrane, we can see lipopolysaccharide projecting outside or projecting from the surface, no, or from the surface to the ambient. Okay, so what are the components? We have the O antigen component. Okay, so that O antigen component here, represented by the blue-purple hexoses, are varied depending on the O antigen. So this is the O antigen that we use to serotype particular species of gram-negatives possessing LPS. No, ito yung varied, right? The inner and the outer core usually are stable, conserved components. And then, of course, the lipid A molecule, the root molecule where it is conjugated to the sugar hexoses are polymer. Sugar polymer is attached to. Lipid A serves as the attachment of the LPS to the outer membrane. Okay. Right, so an example here, the O antigen variations or variants of P. aeruginosa. No? So from 1 to 20, O1 to O20, based on their LPS. You can see different patterns, no? So very different makeup for these serotypes. Okay, right. So how is polysaccharide synthesized in bacteria? Uh, there are many studies on there are many studies on this already, but what I will be focusing on or emphasizing is the most universally conserved, I would say, most universally conserved pathway for the synthesis of polysaccharides. So the WZX WZY dependent pathway. Okay, so there are several molecules here that are involved: WZX, WZY, WZZ, and WAAL. Okay, so here let's set the stage. So there are two spaces or three spaces involved: the cytoplasm, the periplasm, and the ambient. The process begins in the cytoplasm where there's lipid sugar conjugation. No? Then that lipid sugar conjugate, in order for the remaining polymers to be attached to the sugar component, a flipase enzyme works. That's WZX. So WZX flips over the sugar lipid conjugate to the periplasmic space. In the periplasmic space, there are two important polymer enzymes or polymerase enzymes. WZY is the primary polymerase. It extends the sugar component, the sugar polymer, then controls as well. Actually, it's more of the function of the copolymer or the copolymerase, WZZ, controls the final length of the sugar molecule. So more hexoses, sugar units, sugar chairs are added to the polymer, to the growing lipopolysaccharide. And then eventually, okay, we have the action of a ligase enzyme. The ligase WAAL completes the process of O antigen ligation to the lipid A core oligosaccharide molecule that's been finally synthesized by the polymerases WZY and WZZ. So QAAL, WAAL, is a ligase. No? So once that the complete assembly has been, the complete molecule has been assembled, your lipopolysaccharide with the O antigen, it's now ready to be transported to the outer membrane where it can serve its function. And you remember from our module 1, I think I mentioned about the LPT pathway. So that's important for the transfer of or the translocation of the molecule of the lipopolysaccharide to the outer membrane. Okay. All right. Okay, just a reiteration of the processes or the functions of polysaccharides. So complex glycolipids serve as coating materials. No? Very important for forming contact, for signaling, for molecule recognition, and for protection. Okay. Okay, so that ends our part one recorded session. So make sure to watch this before taking our long exam tomorrow.